Dear students, welcome to today's lecture on thallus organization and nutrition in fungi. So we all know the thallus in case of fungi is mostly the mycelial form, where the mycelia is the interwoven mass of the cells, and this interwoven mass of the cells, which we call it as the hyphae. So when you see the thallus organization, except the unicellular forms, is the fungal body is called the mycelium. The mycelium is interwoven mass of the thread-like hyphae, and the hypha may be septate or aseptate, and it may be dimorphic, found in both unicellular and mycelial forms like Candida albicans. The thallus may be of two types, unicellular and multicellular and filamentous. In lower fungi, thallus is more or less single cell structure, and the type of reproduction it becomes the reproductive unit. Such fungi are called holocarpic. In unicellular holocarpic forms, the mycelia is absent, syncytium. In some holocarpic fungi, produces bud cells in succession, and these remain attached to one another in a chain, and such chain of buds are called the pseudomycelia. In the filamentous thallus, this fungi is composed of hyphae, which are collectively aggregate, and they form network is known as the mycelium. Each hyphae may vary in shapes and size. Branch of the hyphae is dichotomous. On the basis of the presence or absence of the septa, they may be non-septate or aseptate or septate. The aseptate hyphae, in this numerous nuclei are present with common mass of cytoplasm without cross walls in the hyphae. Example in oomycetes and zygomycetes, its condition is known as sinusitic. However, septa may be laid down at the time of formation of the reproductive organs to delimit from the rest of the vegetative hyphae, as in pseudosepta. Septate hyphae are septate, and hypha segments may contain one or two nuclei, as case of ascomycotina and basidiomycotina and deuteromycotina. The septa is of two types. One is known as the primary septa. The primary septa are formed in association with mitotic or meiotic nuclear division, and they separate the daughter nuclei. These types of the septa are found in ascomycotina, basidiomycotina, and their asexual state. Adventitious septa are formed in the absence of mitosis or meiosis and occurs especially in association with change in the local concentrations of the cytoplasm. They are found in lower groups of the fungi like mastigomycotina and zygomycotina. When you see the mycelia, there are different types of the mycelium. One is known as the plectenchyma. In this, the hyphae is organized loosely or compactly woven to form a tissue called plectenchyma. And this is again of two types. One is known as the prosenchyma, and the other is known as a pseudoparenchyma. The prosenchyma or prosoplectenchyma, the hyphae are loosely interwoven and are lying more or less parallel to each other. So this is the prosenchyma. And pseudoparenchyma, in this the fungal tissue hyphae are compactly interwoven and they look like the parenchyma in the cross sections. The other type of the mycelia, we call it as the sclerotia. They are hard, dormant bodies consisting of compact hyphae protected by external thickened hyphae. Sclerotium germinate into a mycelia and return to favorable condition. Then comes the rhizomorphs. They are root-like compact interwoven hyphae with distinct growing tails, helps in absorption of the and perennation. Then comes the rhizoids. These are short, root-like filamentous branching of the thallus, found in the tufts of the base of the thallus. They are also found in anchoring and absorbing. Another type of the mycelium we call as Aprisoria, which is singular, we call it as Aprisorium. It is terminal, simple, lobe swollen, mucilaginous structures infecting hyphae which adheres to the surface of the host, and these are formed by parasitic fungi such as powdery mildew and rust. Then the other type is the Hostoria. It is an organ that is developed by a hyphae, usually performing the function of absorption. This is the characteristics of the obligate parasites. They vary in shape and may be knob-like or the button-shaped, elongated finger-like, branched, and they secrete some specific enzymes which hydrolyze the protein and carbohydrates for the host plant. There are also another type of the fungal that is known as the what you call the hyphal traps, which we call it as the snares. These predaceous fungi develop sticky hyphae or the network of hyphal loops known as hyphal traps or snares, which help in capturing the nematodes. The fourth is known as the stromata, which are compact structures much like the matrices, fructification and formed or in them. Regarding the nutrition, the fungi basically are achlorophylous. They cannot synthesize their own food, so they get their nutrition by following ways. One, we call it as the saprotrophs. So saprotrophs are the fungi which obtain their food from the dead decaying organic matter. These fungi secrete certain enzymes outside after digest. Digest the substratum and then absorb the nutrients. The parasitic forms, these are obtained from food from the living organism. They may be facultative or obligate. Facultative parasite grow on variety of the tissues like astilago. The obligate parasite grow only on suitable hosts. Downy mildews are the example. The parasitic fungi that grow on the surface of host cell and absorb food through hysteria are called ecoparasites or ectophytic parasites. Mucor erysiphe. When parasitic fungi grow inside the host tissue, they are called endoparasite and endophytic parasites like Pythium and Puccinia. The other type of the nutrition is called predaceous. Some fungi develop ring-like noses to trap the annelids or the nematodes, anthrobites, zoophagous and dactylia. The other is mutualistic nutrition, where engage in beneficial relationship like mycorrhizae fungi enhance plant growth, lichens involve fungi and algae in symbiosis. The most common types of the mycorrhiza are the ectomycorrhiza and the endomycorrhiza, and also known as, where the endomycorrhiza are known as the arbuscular mycorrhiza. So these are the questions related to the lecture on the nutrition and thallus organization of the fungi, where you have to write a note on thallus organization in the fungi and also describe the mode of nutrition in fungi. So, and the references, if you see, they have been, the presentations are developed from Vashishta and Sinha Botany for degree students. Microbe notes are being considered, online biology notes have been taken into consideration, and biology discussions are being considered. Thank you students for your